Hello everyone, this is Sir ML and please subscribe to my channel. In today's video, I will be taking a closer look into the process of spermiogenesis. Spermiogenesis is the physiological maturation of a spermatid into a spermatozoa, essentially a non-motile and immature spermatid will undergo various physiological changes or transformation and become a motile and mature spermatozoa. So here we have a spermatid which is haploid. It has its rounded nucleus, its two centrioles, its Golgi complex and mitochondria, besides other cell organelles that I did not draw here. So in the first step, there will be changes in the nucleus. The nucleus loses a lot of water and shrinks to assume this somewhat ovoid or oval shaped structure. The RNA content of the nucleus as well as the nucleolus becomes greatly reduced or discarded. The DNA becomes more concentrated and the chromatin material also becomes closely packed into this very small volume. So we can see here the nucleus becomes a little more oval in shape and becomes more condensed. The Golgi complex of the spermatid will then move and become more concentrated towards the anterior end of the spermatid nucleus. One or two vacuoles of the Golgi complex becomes large and occupy the spaces between the tubules of the Golgi complex, and soon after a dense granule is formed which is referred to as the pro-acrosomal granule. This granule will attach itself to the anterior end of the nucleus and enlarges into the acrosome. The mitochondria of the spermatid will also become more concentrated towards the posterior region just behind the nucleus. Similarly, the centrioles of the spermatids becomes arranged one after the other behind the nucleus, as we can see here. These centrioles will then move and become concentrated, or rather become positioned one after the other just behind the nucleus of the spermatid. The anterior centriole is referred to as the proximal centriole, while the posterior centriole, or the one behind the first centriole, is known as the distal centriole. The distal centriole will then change into basal bodies that will give rise to this axoneme or axial filament. And as we can see, a lot of the cytoplasm is also being condensed and being constricted and assumes this shape where the cytoplasm is more and more constricted, and finally excess cytoplasm will be sloughed off as the residual matter. At the same time, the rest of the Golgi complex will also be discarded along with the superfluous material within the cytoplasm, which includes other cell organelles that is not anymore required, and these will all be discarded together so as to reduce the weight of the developing spermatozoa. So we can see here the mitochondria will come and lie behind the distal centriole, and these mitochondria will then fuse together and become twisted spirally around the axial filament to form the middle piece. So as we can see, during the process of spermiogenesis, there will be changes in the nucleus, there will be changes in the cytoplasm, there will be changes in the Golgi complex. The mitochondria will migrate behind the nucleus. The centrioles will also migrate to the posterior region of the nucleus of the spermatid. A lot of the excess cytoplasm and other cell organelles will also be discarded and sloughed off as a residual matter, including the discarded Golgi complex. As we proceed to the later stages of spermiogenesis, the axial filament becomes elongated, and finally the plasma membrane of the sperm will then extend to the tip of the axial filament. And here we can see the figure of a mature spermatozoa that has undergone the process of spermiogenesis. So the pro-acrosomal granule of the Golgi complex will form this acrosome or acrosomal cap that will contain hydrolytic enzymes that will aid the spermatozoa in digesting or breaking up the vitelline membrane of the egg. Behind the acrosomal cap or acrosome will be the nucleus. These two are present in the head of the sperm. Behind the head of the spermatozoa is the neck, where both the proximal centriole and distal centriole are positioned. In the middle piece we have the mitochondria that is spirally wrapped or twisted around the axial filament, and behind the middle piece we have the extending axial filament, as well as the plasma membrane of the sperm, which extends to the tip of the axial filament to form the spermatozoa tail. And that is all about the process of spermiogenesis, or the transformation of spermatid into a mature and motile spermatozoa. I hope you found this video helpful, and if you do, please hit that like button and don't forget to subscribe to my channel. Thank you.